Today I'm going to be talking about vectors. I'm going to be talking about the main four properties of vectors. I'm going to be talking about the roles in making recombinant DNA. I'm going to be talking about the use of phage vectors, viral vectors, and kind of their relationship. And then I'm also going to be talking about plasmid vectors as well as the overall just use of vectors in biotech. So when we're actually looking at recombination within vectors, there is kind of a prior step that I'm missing here, which is called insert generation. And essentially that is when you are trying to isolate your gene of interest, whether this is done through PCR. PCR is obviously when you're trying to amplify, your gene of interest, or just you're amplifying millions of copies of DNA. So it could be done through that, or it could be done through something called restriction digestion, which is kind of what I'm talking about in the first step. And essentially restriction digestion is when you are just cutting out the donor DNA at the restriction sites using restriction enzymes. And that's my step one. So your step one is using those restriction enzymes, cutting at those restriction sites, and then taking that gene and inserting it into your vector, which leads me to my step two. Step two would be to cut open the target vector using those same restriction enzymes. So those restriction enzymes you used in step one, you're going to use those to cut out your vector in step two. And then in step three, you're going to insert your target or your gene of interest into the vector. So in step two, you create blunt or sticky ends, which is going to allow the gene of interest to kind of fit into the vector. And then ligase is going to come in and it's going to seal it up. So once the gene of interest is inserted into your vector, step four would be to transfer that to a host. Now that your vector is carrying in your gene, it can be transferred via transformation, transduction, transfection. And so the four main properties of vectors, what actually makes a vector is number one, you have your origin of replication. So your origin of replication is obviously where DNA is going to replicate, but overall what I mean by this is that they are easy to replicate, that they're easy to clone. And so for plasmids, one of the main things for plasmids, one of the main characteristics of plasmids, especially plasmid vectors, is that they're able to independently replicate. And then whereas phage vectors, you would obviously need a host to replicate. So overall, just making sure that replication is very easy. The other thing that we have are multiple cloning sites. Multiple cloning sites are actually sites with a bunch of restriction sites where your gene of interest is actually going to get inserted. So that's all that means. The multiple cloning site is where your gene of interest is getting inserted. You have selectable markers. Selectable markers are how scientists and researchers determine whether or not a cell has taken up your vector or plasmid. Usually selectable markers have genes that code for antibiotic resistance, and the main way that you could see if your cell has taken up a plasmid is if you were to plate your bacteria on a agar plate or a growth medium containing that antibiotic. If you had your selectable marker and it contained a gene that coded for ampicillin resistance, and you took your bacteria and you plated it on a agar with ampicillin, what would happen is hopefully your bacteria would grow because one, it has ampicillin resistance, and two, you would be able to see that and say, okay, these cells have taken up the vector. So that's all that means. Selectable markers are something that scientists use as a means to determine whether the bacteria or the cell has taken up a vector. The last kind of characteristic is that they're small and easy to transfer. So similar to plasmids, they are small, they're easy to clone, and they are super easy to transport. And the last kind of thing that I want to talk about are viral vectors and phage vectors. So viral vectors is a little bit more broad in terms of that category. It's like it is the category. Viral vectors is a little bit more broad. But what I mean by viral vectors are any type of vector that you're using within eukaryotic cells, especially for like gene therapy and vaccines. Viral vectors are mainly used in like animals, plants, and humans. Essentially how these work is scientists will take a virus, they will remove and modify and take out all the harmful ingredients, all the harmful characteristics of a virus, and they will take a vector and insert it into the virus, transport it to a host organism. That virus will then kind of multiply, replicate within the organism, therefore spreading the gene of interest that you want. In terms of vaccines, typically these viral vectors contain genetic codes for an antigen. It really just depends on what kind of vaccine you're actually using it for. That could be kind of case by case. And then obviously, like I said, they're great for gene therapy. So similar to viral vectors, there are bacteriophages. Bacteriophages are specific phages that will target bacteria. And so vectors can be transported through bacteriophages. It's just they're very limited to bacteria, whereas these viral vectors are good for eukaryotic cells.